Dr. Gail Andrew received her medical and pediatric training at McGill University. As a developmental pediatrician, she has worked in childhood disability. Currently, she is a clinical professor of pediatrics at the University of Alberta and the medical director of the Glenrose Rehabilitative Hospital FASD Clinical Services in Edmonton. This role includes diagnosis, training, education, and research on FASD. She is consulted to the Alberta Cross Ministries Committee on FASD and is on the board of directors of the Canada Northwest FASD Research Network. Dr. Andrew was the scientific chair of the Alberta Consensus Conference of FASD across the lifespan from October 7th to the 9th in 2009, and the conference produced the book, Fetal Alcohol Spectrum Disorder, Management and Policy Perspectives. And I now welcome Dr. Gail Andrew.
Thanks very much, Shelley. Good morning, everyone. We've got a lot of information to cover today, and I hope we can make it as interactive as possible by having questions throughout the presentation. The information on the slides and handout, if you print them, is fairly dense. I want you to have that information for future reference, either within your clinical setting, dealing with families who are raising individual children affected by FASD, or adults themselves who have FASD. So I want you to really be more of a listener so that you don't have to take notes, because there is quite a bit of detail in the slides. And this is an update. I had the privilege of presenting what I call FAS 101 a few years ago. So I want to kind of build on where we've come from over the past 5 years. So what my objectives are to kind of take us to what is the scope of the FASD challenge right now in 2011, and what research has informed our current practice. I talk about the basic science of alcohol as a teratogen, and I'll also talk a little bit more about how research has informed our diagnostic process, and also what interventions we put in place to support that individual across their lifespan. We really know this complex disability requires a lot of interaction between community, clinicians, and researchers in order for us to kind of unravel the intricacies of this disability. At the end, I'm going to get a little theoretical, I think, because I see FASD as a very unique disability. And it really does fit into the framework of strengthening lifelong health. And this particular concept has taken off through the Norleen Foundation. So I'll explore a little bit of that framework at the end. So just to review, what is FASD? Fetal alcohol spectrum disorder refers to this broad range of both physical and developmental symptoms that can affect children born to mothers who consume alcohol in pregnancy. And the key word there is may. Alcohol exposure during the pregnancy is a risk factor. It doesn't link directly to a diagnosis. There are many other factors that affect how alcohol will affect the developing fetal cells. I'll talk a little bit more about that later. Fetal alcohol spectrum disorder, it's a broad umbrella term, and if you review the literature, there's many other terms that fall under that umbrella. Full fetal alcohol syndrome, which is the full disability with significant impairment in brain function, the classic face of fetal alcohol syndrome, and growth deficiency. In my particular clinical experience, that we see the face in less than 10% of our population. Partial FAS, individuals have significant brain dysfunction and some of the facial features. Alcohol-related neurodevelopmental disorder, ARND, is a term that was coined by the Institute of Medicine in 1996 to cover those individuals who have significant brain dysfunction but no physical signs of either growth deficiency or the classic FACE. Alcohol-related birth defects, ARBD, is another term from the IOM, and this term just emphasizes that although brain cells are affected by the alcohol exposure and result in developmental disability, other developing fetal cells can also be affected, such as the heart, kidneys, skeletal system, and that's an important consideration as part of the diagnoses. These terms have been taken into the Canadian guidelines that were produced in 2005. We've actually come a long way, even since 2005, in our understanding, so I think we're almost ready to produce the next set of guidelines. The other important concept of FASD is that it is a lifelong disability. And what we do see is this increasing difficulty in the individuals over time. As societal expectations naturally go up, a person with brain damage is unable to make that same trajectory. So we see increasing difficulties over time and actually increasing needs for support systems in the environment in order to set up that individual for success. I always say that we can't do diagnosis in isolation. Diagnosis needs to lead to intervention. Interventions need to be informed by the current best evidence of what is working to support these individuals. And the unique piece with FASD, it needs to be linked to prevention. This is a preventable disability. I work across disability populations. I work extensively with children with autism spectrum disorder, cerebral palsy, other genetic syndromes. What FASD gives us is a unique opportunity. We know the cause, alcohol consumption in pregnancy. We can focus our prevention efforts at either primary, secondary, tertiary levels to try to reduce the use of alcohol in pregnancy, with the ultimate goal being total abstinence, and therefore no fetuses exposed to alcohol. This is our ideal. We need to do a lot more movement to get there, but this is the unique piece of FASD. The working with birth mothers, I've had a great privilege of meeting fabulous birth mothers. I always say that FASD is a diagnosis for two, and we have an ethical responsibility to try to reach out to those birth mothers and provide them with the supports that they need to make a difference in their own lives. We know that why women drink is linked into many biopsychosocial difficulties in their own personal lives. Just listen to their stories, and it really informs what we need to be doing in the whole scope of prevention. It's not so simple just to say don't use alcohol in pregnancy. We have to address all of those other factors that lead to why women drink. So again, that's why I feel FASD is a very unique disability. And we do have an opportunity to actually break a multi-generational cycle by putting in appropriate supports from the point of diagnosis of an affected child throughout their lifespan, to give them an opportunity to break the cycle and not become the parents of the next child with FASD. In order to support individuals with FASD, we must make sure that those interventions are meaningful to that individual. I think we often sit in our clinic spaces and say, these would all be really good recommendations and suggestions for intervention. But if it's not something within the interest and the goals of that person, we really need to include our clients and their caregivers into developing these interventions. There is a framework that does inform interventions for individuals with disability, and that's the International Classification of Function. So function informs what we need to do to help that individual. But the diagnosis specifically to FASD gives us this unique opportunity for prevention. Just quickly going to go through the history of FASD. is this a new disability? The warning about alcohol use in pregnancy is actually mentioned in the Bible. There's evidence in artwork of the face of FASD into the sort of Middle Ages. So it's been there. People have recognized that alcohol can result in differences in development. The first literature that was published was Lemoyne et al. In France in 1969, with the North American first publications by Smith and Jones in 1973 in the United States. There's been since that point in time, there was isolated literature, genetic literature, anatomical literature. This disability did not hit the forefront of sort of medical journals that certainly I read, and I was a medical student in 1973, so I didn't hear about it until into the 80s. So the Institute of Medicine brought forward a group of experts in the field in 1996 to really put the first well-defined consensus reports on this disability. Sterling Claren and Susan Astley developed the 4-digit code, which is the tool for diagnosis that's used in most of the diagnostic centers. It's an objective way of approaching this diagnosis. They developed that in 1997 and updated it in 2004. The United States and Canada developed their guidelines in around 2004, 2005. We founded the Canada Northwest FASD Research Network, and over the past 5 years, that network has really pushed knowledge about prevention from a women's health perspective, diagnosis and intervention. And now out of this, there is now NeuroDevNet, which is a national group working on fetal alcohol spectrum disorder, again, across all pillars of diagnosis, intervention, prevention. You've heard about in the introduction that we did have an Alberta Consensus Conference in 2009. There's a lot of international work going on by Dr. Riley and his group across many countries. So you can see there has been a burst of information provided just really in the past 5 years. So with that burst in research and information, where are we currently in 2011? So I'm just going to highlight some of the data about the scope of this problem. How big is it? The prevalence of FASD in Canada is felt to be 9.1 thousand live births. That is a common disability. 1% of all people potentially are affected and living with this disability. That comes out to 300,000 people. In Alberta, we've estimated that 23,000 Albertans are living with FASD. All of these numbers are a vast underestimate for many reasons. One is we do not have diagnostic capacity across the country. Even though we're very proud in Alberta to have many diagnostic centers throughout the province to support diagnosis and intervention of individuals. We have not got a surveillance system. There's no place that this data is currently being collected, even at a provincial level. And this is, I think, a need to really develop our provincial database and move that forward. The Public Health Agency of Canada has talked about surveillance and screening, but we don't have rigorous screening tools. And without screening tools going through, we can't even use that for surveillance. So this is a vast underestimate of the scope of this problem. This is just, my next slide shows our current diagnostic capacity. This was work through the Canada Northwest FASD Research Network and just published in 2011. So you can see within this fairly limited capacity across the whole country. Alberta and BC and Saskatchewan and the Yukon have the best capacity, but we still have gaps. And the capacity in Ontario at that point was a 0.39 capacity. So that's slots of capacity refers to the number of diagnostic slots per population times 10,000. So we've got a lot of work to do in training to increase capacity. So how much is this costing us? Through the Institute of Health Economics here in Alberta, Thane and Johnson looked at this economic impact. And in Canada, it was estimated that the cost for FASD was $6.2 billion. So the lifetime cost per person with FASD is $1.8 million. But that doesn't include their loss of productivity, what that person could have been. It doesn't take in the cost and the burden on caregivers, which can be significant. And looking at the breakdown of where that cost is: health, 30% of those dollars; education, 24%; social services, 19%, predominantly in children being involved with the child welfare system; corrections, 14%, and other services, 13% of the dollars. Further information on the cost of the lifetime incremental cost per person with FASD is $742,000, and that was in 2009 dollars. So theoretically, Thane and Johnson looked at this number, $742,000, is the maximum amount that we should be spending to prevent one case of FASD. When you think that's not much compared to the cost of that person, their burden across the lifespan. So I guess we should ask ourselves the question, can we really afford not to focus on prevention? The other system that's really, I think, feels the burden of FASD is the child welfare system. Fuchs and Burnside in Manitoba, because they have a very good database system within their child welfare system, have looked carefully at this, and they've looked at data over 2005, 2007, 2008 through their Center of Excellence for Child Welfare. And this is just some of their numbers that they found, that 11% of children in care had already a diagnosis of FASD, with 6% on the wait list for a diagnostic assessment. Of the children in care with disabilities, 34.2% were FASD, and 39% had a dual diagnosis of ADHD as well as FASD. 89% of children with FASD were permanent wards and had entered into the foster care system at a younger age compared to children with other disabilities. They found that children with FASD entered into care earlier. The mean age of 2.5 years, spent 70% of their life in care, which is a significant. They looked at how much this is costing the child welfare system. FASD children in care cost an additional $43 per day per child, in addition to the base rate of $65, or $15,000 more per year than any other child in care. So FASD is expensive. The largest cost, interestingly enough from my perspective, being a physician, was medications. 5-fold more cost of medications than the general population if you were an FASD child in care. The average number of prescriptions was 4.2 in younger children, with 15 for the older age group, 11 to 15 years of age. 60% of the drugs prescribed to children with FASD were central nervous system drugs that were targeting behaviors or dysfunction or moods. They looked also at the educational cost for children with FASD. They required special education. 38.2% of the Manitoba education budget was for children with FASD. But where did those children go? Grade 9 was their average grade attained. So not equipped to go out into meaningful employability in the world. And of all the recommendations, only 50% of children who had FASD were able to actually connect with the services that had been recommended after their diagnoses, because there was simply a lack of capacity within the communities where they lived. The adolescents, the youth in foster care, they have their own story as well. When children who had started early in the foster care system, as they became older, there was often a breakdown in their care placement. The reasons for breakdown was usually over severe behaviors and other issues. so that when they reached an adolescent stage, where we know with typically developing adolescents, they need their family to support them through that naturally rough growing time. These children were then moved to a different placement. And the average placements for youth, when you look back over the lifespan, they'd had over 14 placements in some. That was the average in Fuchs and Burnside study. The mean age when at their last placement was 15, and then they transitioned often to group homes. And within the group home system, they would often have changes of workers. And this is critical, at a point where they need those relationships. They lost key relationships and trust. And looking at how to transition into adult services, many of the transitions were not as anticipatory as they should be. And many of the children, youth, would then enter the adult system without an education, without employability, and with no single advocate to support them through this. One of our recent observations in my clinic at the Glenner's FASD clinic, we are looking at who are our birth mothers. And we haven't finished the analysis, but it's probably going to be about 90% of the birth mothers were children in care themselves, who were adolescents who were transitioned into adulthood. So I think this is a unique opportunity for prevention. We do know who our next birth mothers potentially could be to target for more prevention. How big is this problem in the justice system? There's been a few really rigorous studies. One was done by Locke, Fass, and Conroy in British Columbia. They looked at an inpatient youth forensic ward. 25% met criteria for a diagnosis of FASD. But right now, we don't have that capacity for diagnosis within the justice system, so we don't actually know our current numbers. The Asante group in British Columbia has developed one of the screening tools looking at screening within the youth justice system for FASD based on behaviors. This tool has not been thoroughly validated as yet, but this is a potential tool to get an idea of how many individuals within the justice system have behaviors that could be on the spectrum of FASD. I think there's some positive movement because the Canadian Bar Association in 2011 passed a resolution recognizing that FASD is a really key problem and needing solutions. So we're actually hoping here in Alberta over the next 1 to 2 years to bring forward, similar to our consensus conference on diagnosis, a consensus conference on the justice system for FASD. And hopefully we'll be able to bring together the researchers, experts, and the people working in the field to give us further information. We talk about diversion, but we don't know what is best practice to divert to. What should it look like? So that outlines a bit of the kind of the scope of the problem. I'd like to just talk briefly about where research has informed our status of prevention. The Prevention Network Activating Team of the Canada Northwest FASD Research Network has brought together two streams. One, Robin Thurmeyer from Saskatchewan looked at primary prevention, and Nancy Poole and her group are looking at the women's issues. And if you go on the Canada Northwest website, you can actually link into some of the publications and information. So I'll just do a very, very quick overview. We actually do know who is at risk for having an FASD child. It's all women of childbearing age across all social strata. Simple, takes two things, pregnancy, alcohol. But not so simple. As I mentioned before, why are women drink? There's huge biopsychosocial issues that need to be considered. One of the really important books that's just been published on this in 2011 was on prevention of FASD by Drs. Clarence, Salmon, and Johnson. And it's available through Wiley Blackwell. And really, anybody working in the prevention field, I think this is essential reading. This is just a little summary of some of the findings. When we talk about it being so simple, alcohol pregnancy, we've done a really good job of putting out pamphlets, coasters, posters, the media on why you shouldn't drink. The core messaging has been incorporated into school curriculums. When people go for primary health care maintenance with their physicians, physicians have been provided with the information about discussing harmfulness of alcohol in pregnancy as part of just general health. Other healthcare providers have this information. But with all this public awareness, why have we not made a big dent in prevention? One of the things that we've found is that professionals need to be trained how to give this message. They need to have the tools to do this. It's not easy to ask a young woman on her first visit into a physician's office, have you been using? You need to do this without blame, shame, and being judgmental. You need to be able to do this questioning in a supportive, positive manner to help her with harm reduction, connecting with services. And we found that surveys that we've done, and physicians in particular, that it's not always asked, although they have the knowledge. We're in a project right now here in Alberta to actually train all healthcare providers and other professionals. So we've targeted third-year university level of professions such as pharmacy, medical students, social workers, and so on, to actually bring them information on what is FASD so they're well informed, and training them on how to provide the information. So that's, I think, going to help people feel better about how to ask the question. But what Thurmaier and their group in Saskatchewan found, that raising awareness does not necessarily change behavior. We have to kind of go into a different layer to make a difference, and that is by training more healthcare providers and community workers around how to do brief interventions, which is spending more time with the person and giving them some strategies. And the next layer would then be to move into motivational interviewing. So those are certainly beyond the scope of my presentation, but just an example of where we need to go to really do a better job in primary prevention. One of the other reasons that primary prevention has not had the huge impact is about half of pregnancies are unplanned. So even though if you have the knowledge that alcohol should not be used during pregnancy, if you're not planning pregnancy and you don't realize you're pregnant till the second month, there's that period of 8 weeks in early gestation where potentially there can be a teratogen exposure. Suzanne Tuffin has done extensive work in this area, and she found that 50% of first-time mothers reported drinking before they knew they were pregnant, mainly low levels of 2 drinks per occasion. But 25% reported binge drinking, and these were some of the younger moms. And binge drinking was defined as more than 5 drinks in 24 hours. And this is disturbing, that after recognizing pregnancy, 18% of first-time mothers continued to drink. So I think that says either we're not, as healthcare providers, supporting them, or they're simply not even asking us for support or even accessing healthcare. And when we looked at some of our inner city women and asked them the question, why are you not accessing healthcare? They said, because when I go, I am not treated with respect, and why would I go back? So I think we have to look closely at how we deliver services to some of our marginalized population. The binge drinking age group is the late adolescents, 15 to 19 years. So it's the same group as who have those unplanned pregnancies. And that's the party culture, university, college students, of social drinking. Military is another high-risk population. And also, I've had some birth moms who've been, high corporate positions, older, not planning pregnancy. So again, it can be anybody. Some of the barriers for women really benefiting from prevention early on is if they're in a partnership with somebody who's continuing to use. It's very hard for her to change. Often they're in abusive relationships. Who are the women most at risk? And this is a study that's been repeated over and over again. And again, I've said, listen to their stories. The women most at risk for having pregnancies exposed to alcohol, they often have unrecognized mental health problems themselves. Depression is anxiety, very common. Living in poverty, lack of educational opportunities to raise themselves out of the poverty situation. And in our First Nations people, the impact of the residential school system. And if people were at the Alberta FASD conference last week, Dr. Martin Brokenleg stressed this intergenerational trauma that still needs to have resolution. The impact of child abuse, again, the whole effect of trauma I'll discuss a little bit later. Women who are raised in the foster care system themselves as children without that support during adolescence and transition to adulthood, at high risk for using substances, experiencing domestic violence. Most of our birth mothers are also very isolated. They don't have support systems. They don't have a best friend. And that's where our support programs, such as the PCAP model, that's what the mentors do. They are able to befriend the women who are on their caseload and guide them into accessing services. Without that kind of support, many of our women cannot. They are willing but cannot change their current life situation. Many of our birth moms are FASD themselves, often undiagnosed. And we are fortunate to have an adult clinic at the Glenrose, and we are able to connect now some of our birth mothers who come to the clinic with their own children to access adult services. Again, intergenerational, trying to break that generational gap. We do have some good systems of care for FASD prevention for the target population, so that's moving beyond primary prevention. And there's many good examples. Shiway program in British Columbia, Breaking the Cycle in Ontario, our first steps, and step by step here in Alberta. And really what they do is they look at those biopsychosocial determinants of health, and they address, try to help with better housing, leave an abusive relationship, deal with their own legal issues, help with parenting, accessing health care. If you had your mentor or a friend take you to an appointment and advocated for you, you will be able to access better health care. And all of these programs, they focus on the brief interventions, motivational interviewing, really developing a support system for these women. And these programs have been well researched by Therese Grant in the States and Carmen Rasmussen here in Alberta. We know these all have positive outcomes. But our big question when we looked at the Alberta experience, when we evaluated where the women were at the point of after the 3 years, many of them had not been able to maintain. So I think I throw this question out. Is 3 years enough? Yes, I think we have a question.
Yes, we have a question from the audience, Gail, wondering what kind of information is being offered to physicians who are the primary caregivers for pregnant women about FASD and the training related to working with these women. That's what the question is about.
Yes, the question from the audience was, what are we currently doing in training physicians to how to ask the question and how to support the women? We've just started this new emphasis on reaching to the third-year medical students. And why we chose the medical students and also third-year professionals. There's still, when you're a medical student, you haven't really made your decision where you're going yet. So the third-year students could be going into family medicine, could be going into obstetric gynecology, could be going into different subspecialties. And we're giving them the program. It's it's fairly rigorous. It can either be a half-day program or a full-day program on everything you need to know as a healthcare provider about FASD, and then how to ask the question. It's a relatively new program. We previously had done, this goes back about 10 years ago, going across the province, meeting with family physicians in their monthly meetings and doing this type of work with them. We have not done that kind of contact throughout the province. That's something that could, I think, would certainly be resurrected to do again. We developed a tool to help physicians ask not just the question of alcohol use, but also asking about domestic violence and nutrition and substance abuse through work with the Alberta Cross Ministries and the Alberta Medical Association a few years ago. And unfortunately, that tool has not had uptake into primary care. One of the problems that we were told with this tool that it took more than 10 minutes to administer. I think as we're moving throughout the province to primary healthcare networks, it doesn't need to be the physician. I think one of the other healthcare providers within that network could be trained to be the person who does that type of interview using this type of tool. And what the tool does is guide you how to ask the question, but much more important, on the flip side of the tool was all the resources you need. What happens if she says yes? What do I need to do next? How do I do the motivational interviewing? And you need to know about the First Steps program. You need to know about addictions services. You need to then know about women's shelters. You need to know about how to connect with mental health services. So I think we still have a lot of training to do, and I think we need to get this back out into the forefront. So I'm quickly going to shift into now some fairly basic science research that has been done over again, emphasizing the past 5 years especially. We've known that alcohol is a teratogen, and we can replicate alcohol brain damage in rat models, mice models, multiple animals in the lab. It is definite brain damage. It's unique. Other substances of abuse, cocaine, crystal meth, does not produce what we see with alcohol. Alcohol is water soluble, crosses directly to the placenta, and targets the developing fetal cells. The cells that are mostly affected are the ones that are at a critical stage of development at that point of time. And alcohol can affect all organ systems. And I'm just going to, this is in your slides as well. This is what fetal development looks like across the 9 months of pregnancy. And within the slide, it shows that different, the heart is developing at different times. Cleft lip and upper lip are affected at different times. Eyes are developing at different times. So each hit of alcohol, that system that's more critically developing at that time, that's where you're going to get the hit. For example, the face of FASD, we only see if there is alcohol consumption for a small 3-day window, 19, 20, 21, roughly, of pregnancy in the humans. So it's a 3-day window. So if there's no alcohol used at that 3-day window, you do not get the face. The face is developed because of blunting of the fetal forebrain development, and the forebrain affects how your facial bones come together later on in development. The brain develops throughout pregnancy. So at any point, you can have the brain cells affected by alcohol in many different ways. And the brain is also developing extensively in the first year of life. But there's not a direct linear relation. The amount of alcohol equates to the amount of damage. We know that the amount of damage can depend on multiple complex factors. One is simply the amount of alcohol and the pattern of alcohol use. We know that binge drinking is one of the most damaging because you reach a very high blood alcohol level in the mother, crosses placenta, high blood alcohol in the fetus, high brain alcohol in the fetus, more likely to have severe damage. The time in gestation, brain developing throughout, so brain is so vulnerable. But we know that in twin studies, not all twins are equally affected. So the fetus brings their own unique genetics and epigenetics to the how the alcohol affects them. The mothers also bring their own genetics into the equation. We know that some enzymes that are involved in alcohol metabolism, acetaldehyde dehydrogenase enzyme in mothers, there's different alleles, different types. And there's two particular alleles. If the mother is carrying them, they actually develop flushing syndrome when they drink. So usually those women choose not to drink because it's not very pleasant. Protective. Is there one of the other alleles, if the women drink because they're metabolizing it relatively quickly, they don't even feel it. So they can, they can drink the lumberjack under the table. And so that particular genetic allele makes her more vulnerable to be a user, an abuser, and to damage her fetus if she doesn't recognize the pregnancy. The maternal nutrition plays a role as well. There is some work in both the laboratory animals and now looking at humans that choline is a common nutrient, may have some sparing effect to the damage of alcohol. Zinc is another potential, it's a coenzyme of many systems that might be potentially protective. I mean, this is, very basic science research. We're not there yet. And I don't think we really are going, looking at nutrition being what's going to fix this disability population. I think it's that prevention that we need to get that across. Talk a little bit what are epigenetic factors, try to explain that to the non-medical people in the audience. Our characteristics are expressed through our DNA, our chromosomes. Epigenetic factors what that is that the DNA itself is not changed, but how our genes are expressed is changed. It's almost like putting a block on an important gene. And these epigenetic factors are controlled from the environment. And there's various biochemical ways that this happens. But if your gene is blocked, then you're not going to be able to either use that gene to produce a protein, an enzyme, so it will have a detrimental effect. And in the rat model, Dr. Joanne Weinberg and her group are looking at the mother rats exposed to alcohol. Alcohol actually changes this methylation. It creates blocking. The other really exciting information from Dr. Weinberg's lab is something that we potentially can control. In the maternal rats, if they're exposed to stress during pregnancy, stress plus alcohol is more damaging. So this whole stress factor is the kind of compounder. And I think that's, again, it's something that we may be able to have some level of control over. And I'll talk a little bit more about the stress factor in a minute. The mother is not the only person undergoing stress. The fetus experiences her stress. And what happens if once you're born, you're in a less than supportive environment, ongoing stress. So I think we need to really focus on the compounding effects of stress. So I'll talk a little bit more about the stress factor. The stress factor comes from, I think there's another question.
Yes, there's a question, Dr. Andrew. Someone is wondering if there's a difference in the response to alcohol between male and female fetuses.
That has not been certainly observed in the human population. I go back to Dr. Weinberg's work. She's shown that the male rats are slightly more vulnerable. With my general childhood disability hat on, we know that male fetuses and newborns are also more at risk from being from at birth asphyxia. So there is some male vulnerability. We don't know what it is, but it's not unique to alcohol. It's any perinatal insult seems to be harder on the male fetus. So maybe I'll talk a little bit about the stress factor. What happens when a mother is experiencing stress? We have our stress system involves part of the brain, the hypothalamic pituitary axis. So there's a signal when you're under stress that the hypothalamus signals the pituitary, and it then signals your adrenal gland. And your adrenal gland is your stress hormone producer, cortisol. So when you're under stress, cortisol pumps up. You have choices. You either fight or flight. When you're chronically in this cycle, your HPA axis is primed. So you're always in this mode. And so your cortisol is high. Cortisol goes right across to the fetus. It's water soluble. Cortisol is on its own can affect the brain cell development. So if you've got cortisol and alcohol, you're very primed to have difficulties not only in fetal brain development, but also how you respond to stressors in your environment later on. And I'll expand a little bit more on that. But we know that maternal stress can be a compounding factor. And that's potentially something we can do something about. So going a little bit more on kind of the animal model, and we've one of the foremost people who started this research was Dr. Kathy Sulik, who has very elegant models of alcohol brain damage. And she's shown that the alcohol affects it can actually kill off the cell, the developing brain cell. It can affect how that cell matures, how that cell migrates to different levels of the brain, and how the cell, once it gets there, organizes. So it's multiple levels of brain damage. And she's also shown that in her mouse strains that there's some particular susceptibility, certain genetic strains. She has a sonic hedgehog gene regulates embryonic development, and certain different variants of the sonic hedgehog have different expressions of the face of FAS. One of our Canadian researchers, Dr. Brian Creans, has shown that alcohol actually affects oxygen species within the developing cell and directly damages the powerhouse of the cell called the mitochondria, and it can deplete the energy. So again, that cell either simply dies or fails to go on and mature as expected. So the animals are telling us lots of things. We already talked a little bit about the epigenetic changes that caused by blocking the DNA. And potentially they can be reversible. But one thing we're finding with these epigenetic changes, they may not be reversible for a few generations. So there's potential that an environmental change blocking a gene can then be passed on to other generations. So this is kind of stay tuned. We need to see where this research is going to see what we can do by controlling the environment. And we've always talked about in FASD that where we need to we can't change the individual, we need to change the environment. But by changing the environment, we can change the individual's epigenetics. So I think we're looking to seeing where this is going in our own particular research that we're doing through NeuroDevNet. We're actually doing epigenetic samples on all the children who come through our study group. It's a very simple test. You just do a Q-tip and swab on the side of the cheek, the inside of the cheek, called a buccal smear. We also get the kids to spit in a test tube. They love it. And we are collecting their cortisol, their stress levels, to help us learn a bit more. So I think my next slide was just more elaboration on the hypothalamic pituitary axis. I want to get into the humans, because that's really where we need to focus. Animals, we can control in the lab, and we can put X amount of alcohol in, we can put X amount of stress in, and then measure all the variables. Our human experiment is so much more complex. It's not so simple. But what we're finding in this early work on humans, we're actually finding that infants prenatally exposed to alcohol, when we look at their cortisol levels, and this is work from BC as well, their cortisol levels are much higher. And also that they don't go through the natural change. If you have some stress, as in the newborn period, cortisol will respond to the stress and go down to basal levels. In our infants with higher cortisol at birth, they don't do this fluctuation. In other words, they're in a chronic stress mode. And this is some new work by Dr. Oberlander from also UBC. He looked at some of the pathways, the neurotransmitter pathways, and 5-HT, that's the serotonin pathway. It's altered in children, in newborn infants prenatally exposed to alcohol. And children who have this type of blunting of the 5-HT, they don't respond to pain and stress in the same way. So we reflect on our newborn infants who have this disorganized pattern of not responding appropriately to just simple environmental stimuli. Because his work is showing that we've got altered pathways, we've got altered cortisol. And he's done some nice work with using the Brazelton scale, where we can show nice normal responses in the newborn period. And our infants prenatally exposed to alcohol have really terrible scale responses. So it's beginning to understand that alcohol and stress combined is causing our much more difficulties than either alone. Another question?
Yes, we do have another question. Somebody is wondering whether you have some thoughts about medications that could be used to alter cortisol levels, either with the mother prenatally or with the infant after birth.
At this point, there haven't been any trials, certainly human trials. I'd probably defer to Dr. Weinberg's lab about what she's looking at within her rat population. But we'd have to be careful, because some of those medications can have side effects on their own. So I think that's another little piece of research when we do the next 5-year update. Hopefully we'll have some of this is relatively new work, but we might have some further information. I'm going to kind of spend some time on neuroimaging studies. So we've got what the rat lab informs us. We've got some early now good work on stressors and the effect of the cortisol on humans. What does this look like in the brain and the parts of the brain that are that we can sample from the periphery? One of the, this is the work of Dr. James Reynolds, also from Queen's University, and our team here, Dr. Carmen Rasmussen is our research lead here at the University of Alberta, and looking at saccadic eye movements. And what saccadic eye movements are rapid eye movements that bring new visual targets onto the center of the retina, where you can, the critical part to see clearly. And these movements are either your voluntary control or automatic in response to a stimulus that suddenly appears. And we know that this is, it's well known about the circuitry. It's controlled by the frontal cortex and basal ganglia brainstem center, so it's a complex circuitry. And as this is a computer test, the child looks at a screen and we measure with this little dot how it moves and how their eyes move. And so it's relatively simple to do. And we find kids with FASD have elongated reaction times, they have excessive errors, and they no express saccadic movements. So really what this is, they can't inhibit. So if you when you're looking at children in the classroom with FASD, they can't inhibit. They're making so many errors, they're impulsive. So this is actually, we can measure this hardwiring by a simple eye movement. This is not diagnostic, but we're learning more about the brain pathways through this particular response. These are some of the early brains from Dr. Claren's first dissections of an infant who succumbed. And they found cerebral cortex, white matter, deep cerebral nuclei, the thalamus, basal ganglia, corpus callosum. They're all vulnerable to alcohol. This is really a gross specimen. And this is the same two brains. Normal brain, child with FASD, lacking convolutions, microcephaly much smaller. It's really rare to see this kind of damage. Usually the damage is microscopic, and it's more of Dr. Claren's work where, instead of having more convolutions, more migration, you end up with a fairly flat looking slice of the brain on microscopic analysis. But we don't want to be doing autopsies and brain biopsies. We need to look. And structural MRIs that we just can order clinically, that I can write a requisition every day, we don't usually see anything in particular. Occasionally, with taking it to a more research level of structural MRI volumetric studies, we see changes in the hippocampus, amygdala, thalamus, caudate, putamen, globus pallidus, and reduced corpus callosum. And that's both our group here under Dr. Beaulieu and Matson have shown that. Dr. Astley from Seattle, Washington, she's looked at the correlation between the size of the frontal lobe and the actual size of the eye. And she's found that there is decrease in the frontal lobe size and volume with a smaller eye. And we know that the eyes are quite a hallmark in the full face of FAS. And the frontal lobe, I talked about the saccadic eye movements, the frontal lobe is really important. The frontal lobe is also critical for executive function deficits, which we'll talk about in a few minutes. Dr. Chudley and his group, very similar. They looked at functional MRIs. It was the prefrontal cortex had less activation when the child was given more complex tasks. So again, executive functioning deficits, frontal lobe. This is a new technique that we've been using called diffusion tensor imaging MRI, looking at measuring the white matter pathways. And it tells us different things. We do volume, but this is, I'll show you this. This is what a DTI looks like. So we look at how water diffuses through the white matter. The white matter is the pathways, and it's wrapped in a fat sheath called myelin sheath. So that's where the water diffusion is different. So we're able to look at how thick the pathways are, and we're also looking at what function is affected by that particular pathway. So we're doing functional testing on the children as well. And what we have found so far with our DTI is 7 out of the 10 tracks we looked at were abnormal. Some of the key tracks, one's called the superior longitudinal fasciculus. Changes in that particular track correlated with reading vocabulary scores. So this is a really important track that connects, again, the frontal part of the brain to the language area of the brain and auditory processing. So a nice correlation with the functional deficit in that brain pathway. We've also found changes in the splenium of the corpus callosum and the left inferior frontal orbital fasciculus. Corpus callosum is a really critical area. It's the big pathway that connects the right side of the brain to the left side of the brain. So underdeveloped pathway. So in terms of processing information, much more slowly. Just recently, Treat and Beaulieu have looked at correlating math ability. And math ability, we know, is a real struggle in our kids with FASD. And it does correlate with differences on DTI in this fractional anisotropy measurement in several regions, especially at the interparietal sulcus. So we're learning function and brain. I think if anybody believes that FASD does not exist, we certainly have the evidence on this research level of brain imaging that really points out that this is real brain damage. And some of the other pictures, I'll just quickly go back to. This particular one shows the difference in the serotonin levels as a result of prenatal exposure to alcohol in the brain. And this is looking just at different sizes in the brain by very elaborate volumetric studies. And certainly, this is Saul's work, that gray matter density was increased. And increasing gray matter density means that you haven't gone through the nice refinement and pruning. You've actually almost got like too many cells. They've not differentiated. And they also found shape distortion in the frontal and parietal lobes. Those frontal lobes again. And this just shows the damage to the thinning of the corpus callosum. So again, lots of evidence that brain damage occurs as a result of prenatal exposure to alcohol. Another question?
Yes, it has to do with the damage caused by alcohol to the brain. So is there a possibility that those things change over lifetime? And if that's true, in what way would the damage change?
The question was, does this damage change over time, and in what way does it change? We have just started repeating DTIs on the same children a few years later, and what we have seen that there is some natural brain maturation, but you've got to remember that typical brains are maturing too. So the underdeveloped areas do change a little bit, but again, there's no catch-up. So the amount of pathway development it doesn't fix over time. Dr. Jackie Pye, her work has been on a lot of work on impact of different interventions, and she has been looking at a program called the MILE program for memory training. So what we are going to be looking at is, can specific interventions make specific changes? So again, 5 years from now, hopefully have more research evidence of can we change areas of the brain by more intensive and specific interventions. But right now, the kids that the few follow-up we've done, we have not, that there's there's natural growth, but it doesn't catch up to the norm. So I think we're going to kind of focus now on the clinical aspects of diagnosis and across the lifespan. I'm going to talk a little bit about screening tools because some members of the audience may have participated in other webinars that were put on by CAFC, the Canadian Association of Pediatric Health Centers. They partnered with the Public Health Agency of Canada to look at some promising screening tools. So that was published. There is a toolkit available on the CAFC website, cafc.org. In 2010. There is a need for further research and validation, so I'll just talk a little briefly about what these screening tools are. One is the neurobehavioral screening tool, and this was derived from the Achenbach Child Behavior Checklist. So they looked at some critical questions about child behavior and found that some of these questions seemed to be more specific for children prenatally exposed to alcohol. This initial work was done by Dr. Korn at Toronto Sick Children's Mother Risk Program. And we are currently, in our research at the Glenrose, looking at doing some validation of this particular tool. And what we have found that, it's a retrospective study, so we administered the tool to children who already have a diagnosis of FASD. And it was 36% of those children scored positive on this screening tool, which means that a higher percentage did not score positive. The tool is very specific, though, because when we administered the tool to controls, we picked up no controls. But what the tool seems to pick up is more sort of conduct disorder, oppositional behaviors. And those are common behaviors in many of our children who have other mental health diagnoses. So what we are now doing is we're going to be doing a prospective study of administering the screening tool to children prior to coming into clinic, and we're going to see if it correlates with their diagnoses. So I think a concern that it's going to pick up all children who have significant behaviors. And we certainly need to pick up all those children, but they're not necessarily going to be prenatally exposed to alcohol. One of the other screening tools is meconium free fatty acid ethyl analysis. Meconium is the first bowel movement of the newborn, and alcohol leads to a specific pattern of these exposure in utero to these free fatty acid ethyl esters, which would be a nice biomarker, but meconium is not produced until about the 13th week of gestation. So drinking heavily in those critical early months of pregnancy and then stopping, you could have big window for brain damage. Your test is negative. So is not reassuring. That newborn is screened, perfectly fine. The other corollary is if there's no alcohol use until maybe 2 weeks prior to delivery, which is still a time where brain's developing, so potential for damage. But that fetus would have a strongly positive test. So there's a lot of ethical issues. The Canadian Association of Pediatric Health Centers did host a symposium with ethicists on this particular subject in September. And I haven't seen the proceedings coming out of that, but I think we need to be well informed by our ethics people before we embark on some of these types of screening tools. One of the other screening tools is this maternal drinking guide. And certainly a confirmation of exposure to alcohol is needed before we can proceed with the diagnoses. But we know that there's all those other complex factors. Maternal drinking is not diagnostic. So if we use a maternal drinking screening tool, that child requires a diagnostic workup. So we need to increase diagnostic capacity if we're using that type of universal screening tool. The medicine wheel screening tool is a very holistic approach to working with the First Nations community around preparing the community for considering this diagnosis and preparing a community to support both the birth mother and the child after. I think it's a wonderful, again, a framework, but it's not really a screening tool. Screening tool that I mentioned briefly that Asante and his group developed for the youth probation officers is looking for challenging behaviors and patterns of brain dysfunction to be used for screening like youth probation officers in the justice system. Again, a lot of work still needs to be done on these screening tools. Currently, the best screen is that confirmed alcohol exposure and a child who's struggling in some way. That's the entry point into a diagnostic center. And we really do need to work on increasing diagnostic capacity after screen positive. Because again, ethically, if a screen is positive and if you don't proceed with a full diagnosis to confirm, you're not doing anyone a favor. And diagnosis is the entry point to intervention. So where are we in terms of diagnoses? Well, we do need the alcohol exposure. How to confirm that alcohol exposure is quite a rigorous process in those clinics who are following the 4-digit code recommended tool for assessment. There's usually that's a major responsibility of the intake coordinator of the clinic to really pursue this. Many sources, notification of live birth records, often it's not recorded, looking at child welfare reports, reports from the justice system, interviewing significant family members. The best is to interview the birth mother, but that requires a lot of respect. No blame and shame, not judgmental, and being there to support her through this process and connecting her to resources. So the history of alcohol exposure can be quite difficult. Looking through child welfare records is another resource. Possibly there's alcohol that the mother discloses during an interview. Have to be careful that somebody doesn't have a vested interest in saying that we saw that mother drink, because if there's like a custody issue or something. So it's a very serious question about asking the alcohol history and finding it. What we do in our clinics is really look for, is there evidence for organic brain damage from the alcohol exposure? And this requires a multidisciplinary team. And what we look at is not just the alcohol exposure. We also look at all the other pre- and postnatal factors. You know, we talked a bit about the maternal stress. What other possible substances did she use during the pregnancy? We look postnatally at the trauma of the family unit, chaotic lifestyle, ongoing substance use by the mother, less than appropriate stimulation, neglect, secondary acquired brain injury from non-accidental trauma or accidental trauma, being born extremely prematurely. So a lot of data is collected, and we carefully consider a differential diagnosis. What else could this be? And the other is the comorbidity. What else is it? Most of the children and youth who leave our clinic don't leave with one diagnosis of FASD. They leave with multiple diagnoses, usually of a mental health nature. I think we've talked a little bit about the complexity in FASD because of trauma. We talked about the prenatal trauma, but postnatal trauma. And I'd like to, the camera can pick this up. It's my show and tell. We don't have time to really talk about trauma, but through the Alberta Family Wellness Initiative, with the Norleen Foundation in partnership with Harvard University, there's wonderful volumes of data on the early brain development. So at the end, I actually have these references at the end of my slides. So these are available online and through the Norleen Foundation free. So if you want to explore the impact of early trauma, I strongly suggest that you access that material. The stories, I keep going back to stories. The stories of my children and youth are sometimes incredible. Multiple home environments, in and out of the foster care system, not being able to attach to a single care provider. It goes on and on. And often these children, and I'll focus a bit on the foster care system, they enter into the system. But unless there is that awareness or knowledge about the prenatal exposure to alcohol, that particular child may not be understood. Their behaviors may be misinterpreted. And their caregivers may not be able to access the right door for the right supports. So that's why I really, I guess, strongly want to emphasize early access to alcohol history, interventions, and supports for child and caregiver. On the other hand, we've actually, the power of environments. We see children who are heavily prenatally exposed to alcohol placed in a very supportive environment, and they often do really well early on. I'm thinking of many of my children who are in adoptive families from birth. They usually don't come to my clinic door till they're about 8 or 11 years of age. And it's that age where they are required then to start using their executive functions, and they don't really have them. What we've learned in our clinic is a need for longitudinal follow-up for these children at risk, because we see more difficulties over time. What the clinic does, it moves beyond the basic tests of IQ, basic language, basic motor skills into more complex testing. We want to see how this brain functions. So that's actually the testing process to look for deficits in executive functioning. Dr. Rasmussen has been one of the leaders in this field of defining the executive functioning deficits, along with many other researchers. So the deficits that we find in FASD is deficits in memory, motor planning, attention, higher level communication, executive functions such as inhibition, shifting, flexible thinking, sequencing, judgment. Those are really the core deficits. So that's what a diagnostic multidisciplinary team needs to be measuring. And how this is expressed, many of the children enter in with this long list of difficulties, learning difficulties, mental health issues, ADHD being the commonest one, neurobehavioral very dysregulated, failing life, not being able to adapt. A lot of these children have already been diagnosed with several other alphabet soups, I guess you call it, ADHD, opposition-defiant disorder, conduct disorder, depression, anxiety, attachment disorder, post-traumatic stress disorder. And many of these diagnoses have been assigned based on, rigorous use of the DSM, behaviors checklist. But the root cause, if you don't have the prenatal exposure to alcohol, you don't really understand where all these other diagnoses fit in the bigger picture. So that's why alcohol history is critical, because it helps not only inform why this child is having all these difficulties, but also helps inform how you might put in interventions. For example, the intervention for a child with depression might be cognitive behavioral therapy, and as well as medications. If you then know you've got prenatal exposure to alcohol as your root cause, and the child has executive functioning deficits, CBT therapy is not going to work. Talk therapy may not work. You're dealing with brain damage. You need to shift your approach. So again, I can't emphasize more strongly the need for that diagnosis of FASD, the knowledge that alcohol is the root cause. We talk about secondary disabilities, and secondary disabilities were thought by Dr. Streisgerth in her early work, and she's followed a cohort of individuals with FASD for now about 35 years. So it was thought to be related to not being diagnosed early, not being understood, being set up for failures because people's expectations were too high, and so on. What we are learning, some of these secondary disabilities are maybe actually related to the primary alcohol damage. The new evidence, serotonin is a really important neurotransmitter. We're finding out that the alcohol exposure and stress in utero changes the serotonin levels. So that might be why we see so much mental health disability in this population. It is actually an expression of that brain damage. And the other is this new compounder of as we know more about the trauma on brain damage. So, and now as we're learning more about epigenetic factors, so we're shifting. Secondary disabilities may be actually primary disabilities. So the secondary disabilities are usually mental health, nature, trouble with the law, not being able to live independently, employment difficulties, and so on. So we've actually shifted away from secondary. This is the compound of that individual. And we always said, theoretically, we could prevent secondary disabilities by early diagnoses and early and supportive environments. Think of that child in that positive adoptive home who's struggling. So these are most likely primary. So our diagnostic process, just quickly spending a few minutes on this, we look at the 4 core areas of growth: face, brain function, alcohol exposure, pre and postnatal factors, a lot of data gathering, and the whole team. Our team is speech language pathologist, occupational therapist, psychologist, pediatrician, and social worker. And we do this whole menu of tests. We do questionnaires to sample across all different environments, home and school and community, and we do a very in-depth mental health assessment. Growth, the only new thing in terms of growth is that there's new growth charts from the World Health Organization. So we look for height and weight below the 10th percentile, but using the new norms. And consider all other causes of small stature, as well as taking the family size into consideration. And also weight can be changed by all of our psychotropic medications. The facial features, face comes with that exposure to 19, 20, 21 of pregnancy. And this is we typical face that Dr. Sulik's, this is her animal model. You can see the bottom one is total blunting of that mid-face development. The upper one is that nice lip and philtrum. And there's the typical face, the small eyes, palpebral fissures, flat philtrum, very thin upper lip. And that's how we measure it, with going from one end of the eye to the other. We also have a photographic analysis and a computer program that does this for us. We've got new norms for the palpebral fissures that were produced by Chudley and Claren through the Canadian populations. Again, we should be using the Canadian norms. And this is how we simply look at the lip and philtrum. It's just with a photographic a picture from 5 to 1. 5 is the full FAS, no philtrum, thin lip, to number 1 being normal. And we have another guide for Afro-American population. And that's, you can see the difference between the lips. But it's really not all about the face. You don't fail kindergarten because of your thin lip. This is the real root of the problem. So we need to go, these are the domains that we assess during diagnosis. The intellectual, the IQ is often well within the normal range, can be scattered, but doesn't really represent the dysfunction. Academic achievement, math difficulties, reading comprehension are the two typical ones. A lot of difficulties with attention, both sustained attention, shifting, and a lot of impulsivity. It's the commonest comorbidity. We have 64% of our children leave with a diagnosis of ADHD as well. Can have difficulty in the sensory motor area. The typical difficulties are motor planning, visual spatial, soft and hard neurological signs, and sensory reactivity. There's some interesting work exploring what is the sensory deficit in FASD. Is it reflecting the brain damage from the alcohol, or is it reflecting the trauma and this cortisol reactivity, or is it a primary sensory deficit from the brain damage? So lots of things to explore when we're looking at the sensory. Sensory is often an area that we can target within the environment for supports as part of a multimodal intervention plan. In communication, what we typically see, difficulties with language formulation, disordered pattern, so that they're very talkative, so use of language is actually better developed than understanding, which is the opposite of normal development. They can't connect when they try to tell stories. They're all over the place, disconnected. And poor perspective taking. Perspective taking means that you actually understand that another person has a different point of view, a different idea, so that you can, when you're talking, you give them more information so that they can understand your perspective. And also that you, when you're listening to them, you look at nonverbal cues and take it all into consideration. This is a serious deficit in many of our kids that leads to not just academic difficulties, but social difficulties, contributes to the poor judgment and being successful. Memory can be hit in many ways: encoding, getting the material in, retrieving it. It can affect visual memory, verbal memory, working memory. Executive functions. In our clinic assessment, we look at inhibition, shifting, judgment, sequencing, organizing, problem solving, mental flexibility, abstract reasoning. So the assessment takes a day and a half, and these are the complex reasons why. We measure adaptive functioning, day-to-day, how they function day-to-day life, measure safety risk as well. And we measure all mental health status, so their behaviors, emotional regulation, attachment, and sleep. So we look at all this information and formulate the diagnosis. And according to the DPN, you have to have 3 areas of difficulty that are not correlated. For example, if your ADHD is not treated and you can't pay attention to getting information in, we would not necessarily assign a diagnosis till we treated the ADHD. We'd then bring that child back for a review assessment, because we need to have 3 different areas of difficulty. Another question?
Yes, there's a question about the assessment process and to what extent the child's home environment is assessed as part of the assessment, so the caregiver environment.
The question was about what about the child's home environment, the caregiver's component. You know, when we do training a diagnostic team, we spend about a half day on that particular topic. Yeah, a caregiver interview is done. It's co-led by the physician on the team and the social worker on the team. And we're looking at a description of how many children are in the home, what strategies. And I'm amazed at some of the strategies caregivers have come up with. I learn more from them every day. What kind of, do they have respite? Does the caregiver understand FASD? Are they committed to the child? So we actually drill down on that. We actually look at how many moves. Has this been a stable placement? We will not see a child unless they've been stable for 6 months. So they can't have a lot of mobility around the time of assessment. We also look at the mental health of the caregiver. And as I mentioned before, some of our birth moms have actually said, I think I have FASD myself. And it certainly then explains why they have never been able to connect with services or to follow through. So it's an in-depth process of trying to get a snapshot and a little bit of a moving picture of that child in real life and across environments, the school environment, and also community safety, recreation, and how are they doing out there. We often give more than one diagnosis, and this is just a breakdown. ADHD, 64% of our children get an ADHD diagnosis. Cognitive impairment, 35%. Mental health diagnosis, anxiety, about 15% of our children. Depression, 8%. Attachment disorder, 15%. PTSD, 12%. Sleep disorder, 35% with sleep disorder. So it's just a little bit of a cross-section of who actually comes to our clinic. So after clinic, though, we really we want to come up with strengths-based focus. And that's what this, it's a day and a half of assessment. We get a lot of information on function and strengths, because that's what we're going to, put our interventions on, strengths, to support them through their deficits. And we developed a fairly lengthy recommendation in discussion with the caregiver, and depending on the age of the child or youth and their capacity to bring them into part of the recommendation process. Recommendations need to focus across all sectors. Education, whether it's early education to employability at the other end of the age span. Medical needs, sleep disorders are huge. Dental care is often horrible. Vision, recommendations for mental health. How is that child going to participate in their community? What's it going to take? Ask what do they need if they're already connected with the legal system. The recommendations need to provide training for caregivers and ultimately that transition into the whole of their adulthood with safe housing, employability, and so on into adulthood. What we find, most of our families, even those who have very strong caregivers, they still need a navigator. This is a complex system because it really is crossing all systems of care. In our clinic, we've developed a short-term, a bit of a case manager support with our social worker, and then a handoff to, once we've got them connected, for example, with coaching families in the community. But this is, we find over and over again, this is what's needed. And especially if our caregiver is a birth mother still struggling with their own issues. So a lot of support and individual care needs to be put in place. And I'm just going to reflect on one of my teenagers last week. We finally built a team around him, and he said, I've got my peeps. He's got his people. So it was quite nice. But that's what's needed. And in the slides, I've shared our recommendation template. So those of you who are working in communities, please feel free to use our recommendation template. I'm going to just go through a little bit how this can present across the lifespan, and looking at the environment. That answering to that other question, how can the environment change the presentation? Newborn and infancy is a very important time to be able to access the prenatal history of alcohol exposure. And my colleagues in obstetrics and who do pediatric neonatal intensive care, they are attuned to look for this information and to at least document that the alcohol exposure was there. Not a very small percentage of children prenatally exposed will present with direct symptoms. Neonatal absence syndrome, NAS, is more usually associated with drug exposure in utero, but some children with the prenatal exposure to alcohol, going back to my cortisol story, they come into the world dysregulated. They don't respond to even the gentle touch that you would hold a newborn appropriately. They're reactive, and they don't calm. They have disorganized suck and swallow, affects their ability to feed, can lead to failure to thrive. So really, there can be symptoms directly at birth. Most kids, there's no symptoms, so when we go to early discharge, 24 hours, you're home with your birth mom, and if she's not got strong supports and doesn't understand this baby, not responding to her, and she's trying her best to bond to her baby, and the baby's, it's a huge setup for failure and potential child abuse. So that's where even if the mother, if we know the alcohol exposure, building her support team right there. And we have that model in the PCAP model, the First Steps model here in Edmonton. Birth to mom supports, optimal time to reach out to her and give her what she needs. If the child is going into a different care environment, we owe it to that caregiver to tell them about the prenatal exposure to alcohol so they can then understand this child's difficult responses, feeding difficulties, and sleep cycles. Because otherwise we're going to have a breakdown in that caregiving situation and another and another and another. So understanding and building in the supports, because often just simply giving them some helps in the sensory systems that maybe you should feed this way, maybe you should just tightly swaddle and not rock, maybe you should dim the lights and soft, no noise, which is just usually the opposite of what we like to rock and swing and sing. So, using that different approach can really be helpful. And if a child has prenatal exposure to alcohol, it's not a diagnosis. I've only made one newborn diagnosis, and that was full face and severe neurological compromise. But that child requires ongoing developmental follow-up and access to early services. So in toddlerhood, there's a whole variety of presentations in toddlerhood. And in this particular age group, hugely affected by the understanding and supportive environment. They may have some delays in development. Sometimes it's not so much a delay, but it's just that disorganized patterns, the qualitative piece more than actually a delay. This is usually where you're going to see the busyness and not being able to focus and attend, flitting from toy to toy. Usually very active. Some can be either over-responsive stimulation or under-responsive, like not really responding to the environment. And in those situations, you're really looking at, is this alcohol or is this postnatal trauma? What is this that's affecting this child's ability to respond to their environment? Because I've seen some children that appear to be completely dissociated and shut down. And in fact, the little guy that I saw in follow-up last week, our question was autism, dissociative disorder, or the impact of the prenatal exposure to alcohol. He was provided with an enriched environment and has just taken off. Wonderful to see, and his environment is very rich. His other environment was not so rich, so the impact, yes, he has alcohol exposure, but what I was seeing was environment, and trauma. It's important to recognize prenatal exposed kids in this particular age group to really work on attachment and prevent this multiple moves and to prevent PTSD. Because these children are difficult, they are sometimes difficult to form an attachment to, and that's why I'm very excited to be part of a program that's under Child and Adolescent Services called the CATCH program. We are going to be working with children from 0 to 5 who are coming into the foster care system. We're going to be working with their caregivers, both birth and foster care providers, to provide this wraparound understanding of the child's underlying factors, the understanding the trauma, their developmental, behavioral, emotional attachment needs. And again, wraparound. So we need wraparound from the cradle to the grave, I think, in this disability. management of sleep disorder is quite important in this particular age group, and looking at positive sleep hygiene environmentally, not medications at this point unless absolutely necessary. So this is often what we can assess at this particular age group are the basic skills. We don't necessarily have evidence of organic brain damage for a definitive diagnosis, but we need to provide them with developmental services to address all their needs. And what are the developmental services? They're good, generic community early intervention programs, working with the caregivers, setting up the environment, and optimal training for their whole team involved. And then these children need to be monitored and brought forward for further assessment when they're older. When getting a little bit more confident in our ability to look for organic brain damage as the child gets older. Kindergarten years, age 5 to 6, we often see more difficulties arising because this is the first group learning experience for many of these children. If they've been in early intervention, they've had more one-on-one, but now they're in this class of 20 plus children, and it's a little overstimulating, so they can really present more with this ADHD pattern at this particular age group. They also have more social difficulties. They can't inhibit their impulsive, they don't read cues, they don't recognize social or safety boundaries. Caregivers often say they simply don't listen. Well, it may be that they're not understanding language. It's not that they're listening, but they're just not getting it. We often see kids labeled oppositional defiant disorder at this very young age. And ODD, that's what the behavior looks like. But if you kind of go back, if it's based on brain damage, they may not necessarily be able to have the intent to be oppositional and defiant. They're not willful. It's that expression we use, they simply can't. They know maybe that they're not supposed to do this, but they're impulsive, they're going to do it anyway. So at this age group, we may be able to tease out more brain damage. The language is a pink flag. They're often very talkative and chatty, tangential, and their understanding is less developed. So basic language testing will pick that up by a speech-language pathologist. Listening to the story of caregivers will often tell you, you see this wonderful child in front of you, and you hear the story of the caregivers and how much support they need to put in place to get through a day. Ask the teacher in kindergarten how much they're putting in place. A huge amount of supports. So at this particular age, we may or may not have evidence for organic brain damage, but we need to continue to support them functionally. Dr. Jackie Pye and I looked at our clinic sample and found that it was we were probably doing a 5-year-old diagnosis probably about 10% of the time. And these were kids that had very definite cognitive impairment along with the behaviors and serious language issues. Most of the children, we do not have sufficient evidence at this age group for a definitive diagnosis, but we still need to support. Medications are often considered at this time, especially for the ADHD component, but medications are for targeted behaviors. This is where we can, by the age of 8, administer all of our executive functioning testing. We can move our motor testing and our speech-language testing into more complex functional skills. And we can really get a really good sense of function. What do caregivers tell us at this age? The kids are no longer keeping up academically. The early grades is basic rote learning. When you hit grade 3, you're supposed to now do something with it. You're supposed to have that cognitive flexibility. So math is usually the first area of struggle that we begin to see, and then the language of learning, not understanding language, reading comprehension. ADHD and ODD, again, very common presentations. Caregivers will often say, my kids can't make or keep friends, they're immature, they don't get social cues. They're the ones that are either bullying or being bullied. Can't organize, can't get through a day without constant supervision, the caregiver breaking down every step of the way. Can't handle unstructured times. Very, very socially inept. Not learning from consequences, very common comment. Impulsive, poor judgment, lying, stealing, in spite of being told over and over again. Well, if you've got impaired memory, if you've got poor impulse control, and you've got poor executive functions, even though you've said that you're going to do this, and this, at the heat of the moment and where other things are going on, you're not going to be able to pull that out of your brain to actually use it. So you're going to lie and steal again. Lots of mood swings and regulation difficulties with motor planning. So those are what the caregivers tell us. What do we see when we bring the child in? That's what we see. And I think our commonest comment when we're giving information back to the caregiver after the assessment is that we'll often say, we're not going to tell you anything new because you told us. In real life, this is what you're looking at. In our assessment, this is where the brain deficit is. So we really can see this correlation of hard measurement of brain testing with where the child's functioning. This particular age group, the 8 to 10, we're looking at what are the best interventions. These children are often in a behavior disorder classroom. This may not be the best place because they're very vulnerable. They copy behaviors. They do best in an environment that's not overstimulating. They need specific things to address their learning difficulties. They often qualify for a learning disability pattern. They need to have ADHD managed by environmental strategies and most likely medications. Mood disorders are very common. Again, medications may be required. We're looking at, Dr. Jackie Pye is looking at specific interventions and strategies for teaching. So hopefully in 5 years we'll have more specific interventions that can help. We need a lot of caregiver support. Adolescence kind of just, this expands on what I talked about, the middle age group. Into adolescence, you really see more of those difficulties in community and functioning in general with mood and behavior regulation difficulties. They simply don't fit in. And that's the brain damage. It's not that they can't, it's not that they won't, it's they can't because of their brain damage. And this is further compromised, as we talked earlier about, if an adolescent has moved and they don't have their support system in place. If they've had disruptive schooling, attachment issues are still involved. If they've not dealt with unresolved trauma, this is where it really hits when into adolescence. And their presentation in adolescence is really this, the mental health is huge. Some of the adolescents that have come through our clinic recently have already been living on the street. They have not been well supported. And what's missing is that significant person in their life. And part of the interventions is to develop that support team, but a lot of our youth don't want it. My buddy who has his peeps, he wanted it. But I have a hard time selling that you need a wraparound support system. Adults just keep going. The inability to function in life is so evident when you meet an adult with FASD. Often they don't necessarily present with any facial features, and it's hard to find the prenatal exposure to alcohol. But in our adult clinic, we are actually doing that full battery of neuropsychological testing to drill down on the brain. And that so informs information about what we need to put in place. Again, that whole wraparound system. And it has to be wraparound home, community, and employment. I'm going to just wrap up by looking at some more specific interventions for FASD and what we've learned again in the past few years. Environment strategies. That cannot be emphasized. And I do my, I guess I should call it my 3 S's: support, supervision, safety. Those are the 3 things that are required. And I wish I could just stop there and say that we could fix everything, but there is a definite role for medications to help that individual cope in their environment. Because going back to the new brain damage, serotonin pathways are kind of wonky. And maybe the cortisol is still not as responsive as it should be. So medications, for targeted, we don't medicate FASD. We medicate for targeted symptoms. ADHD is common, depression, anxiety, sleep. So let's explore a couple of these. ADHD management is very similar to how we manage ADHD in familial type of ADHD. The commonest, the methylphenidate, dextrine stimulant medications. I like to use long-acting forms because, to be fair to the child and family, after school hours of homework, cubs, violin lessons, whatever, and the family life. The family deserves to have some medication still working. I have a hard time getting long-acting medications covered by certain health medication plans, and our First Nations children are also not allowed to receive that medication unless we do a lot of letter writing. The longer-acting ones are really better for the child and family. Not good studies published to date on medications in FASD. A few of the studies have said, well, less robust response to medications. My clinical experience, that's right. Our kids often need higher doses and may need different products tried. Again, brain receptors change. Drugs are geared to a certain receptor. Got to monitor for side effects. A lot of our kids get tics and poor appetite, sleep disorder as a secondary side effect of the medication. And the reason why meds don't work that well in FASD, going back to our brain damage slides, frontal lobe. That's where the medications work. Frontal lobe is when the primary areas of damage or the connecting pathways. But can't say, ADHD meds on their own don't work without environment supports. Need them both. Moods. Moods like anxiety and depression can be secondary disabilities, but now we're moving more their primary disabilities. We can certainly try to prevent the secondary disabilities by setting up the environment. And sometimes I can help a child who's anxious by, if they're having multiple disruptive visits between family members, if they're frustrated at school because they're lost, just changing their learning program will relieve anxiety. Maybe they're being bullied, let's address the bullying. Or if they're attachment issues, let's help them with attachment and trauma, and maybe the moods will go down. Cognitive behavioral therapy typically does not work in this population because you're dealing with brain damage and language difficulties. They often do better by play therapy or alternative therapies. I do use medication, and so do my colleagues in psychiatry. And again, targeting the serotonin pathways, Zoloft and Prozac for depression, Celexa for anxiety. There is a role, but I would really emphasize, again, environment strategies first, medications next. Sleep is a huge issue. My sample, 35% of my kids have sleep disorders. Starting from basic sleep hygiene. Some of my kids are in chaotic environments. They actually share a room with 3 other kids. Or they're allowed to take their DS to bed. Or they're so worried. Child that I worked with this week, he's actually parenting his parent. He is so worried about her health. Well, of course he's not, and he's very anxious. So do I medicate him, or do I put supports in for his mother so he doesn't have to worry? So those are the kind of thinking I think we need to look at, considering all factors. When we look at sleep has many different causes. One is, you have too much on your mind and worried. The other is your airway. Going back to Dr. Sulik's little rats and underdevelopment of the face, that's where your whole breathing mechanism is. If you have flattened through the frontal fetal forebrain, your nose is going to be smaller, and the airway from your nose back is smaller. So when you sleep, what happens? Your airway closes. That's called obstructive sleep apnea, and we can actually measure this in our sleep lab by, you got oxygens that go up and down all night. Well, no wonder you're not getting a decent sleep, and you're restless and turning all around. Management for that is relatively simple. We actually help the airway by taking out the adenoids so there's less to flop back, or in serious cases using a BiPAP machine just to actually blow air into the nose at nighttime. Something fairly simple that can fix this. But the majority of our children don't have that, but I think it's something we need to think about because we can do something very nicely surgically about it. What medications do I use for sleep? Start with something natural and more simple, melatonin. It helps with sleep initiation. It doesn't keep you asleep, but at least it can get a child to sleep. And maybe if you've got the sleep hygiene, like the child now has their own room and it's calm and they have a nice cuddly blanket and a transitional teddy bear, all good to go. When melatonin doesn't work, my own preference, everybody gets their own preference, I move up to clonidine. Is there a role for trazodone and some of the other more sedative medications? I think it's a case-by-case basis. One warning, I do not use risperidone, an antipsychotic for sleep. That unfortunately has become the first drug of choice for many people who don't understand this disability. And I'll talk a little bit more about that in a second. Aggression, how do we manage aggression? ODD, CD, conduct disorder, opposition defiant disorder. Well, take a step back. What is the root cause? If the child has brain damage, if they're cognitively challenged, if they don't understand the language that you're trying to tell them, don't do that, don't do that. If they're sensory reactive, if they've got all those executive function deficits, that can be the root cause of them appearing to be oppositional, of appearing to be very aggressive. And so again, the environment, making sure that the environment understands the child's disability, that they've got the huge level of supports. If the child's aggressive on the playground, maybe having a buddy system for that child on the playground so that they aren't getting overstimulated into the wrong crowd and being torn in that direction. So kind of looking at fairly simple environmental things that might not set up that situation. Lying and stealing. A lot of my caregivers say they just simply sew up the jeans pockets. You can't put in the toy from the dollar store if you've got your pockets sewn. So little strategies may help. So I typically don't use medications for these symptoms. I find that if we look at all the supports, and also dealing with attachment and trauma through therapy. Is there a role for risperidone? You need to really look carefully at side effects versus benefit. When aggression and impulsivity are huge safety issues, this may be the case that this might be the medication that is required after everything else is considered. Side effects can be significant weight gain, and long-term can lead to metabolic syndrome. There's a new product called Abilify that may have less of these side effects. Excellent review of risperidone and atypicals was recently published in Pediatrics and Child Health 2011. So I think we've quickly gone through what's happened in the past 5 years, from a systems community to the lab with the rats, to the neuroimaging studies, to the clinic, and then support systems. So just to wrap up, this really is a system of care problem. It fits with the information coming in from the Norleen Foundation that we really need to look at a framework of policies and programs to strengthen lifelong health. And to break that cycle, it's a real cycle from preconception, prenatal life, early childhood, middle childhood, adolescence, adulthood, and guess what, you're back at the cycle of babies again. So we need to look at it in this cycle, and we need to put at all points in that cycle evidence-based strategies and preventions, and really supporting the caregiver and community and increasing capacity. So I'll end at that point. I think if the sustainability of the system, I don't think it's not an option. It needs to be sustainable. Because going back to my dollars and cents slide, it's expensive if we don't. So I think I'll leave it at that point, and if there's more questions coming in, I'll certainly be able to even electronically respond to some of them.
Yes, there are several questions. There have been a lot of questions throughout. One of the questions that you may have time to answer, we just have time for one question, is what the value would be of diagnosing an adult who has an IQ of over 70, so is able to function in the average range. What would be the advantages of a diagnosis for that person?
I think I would question the ability to function component. The question was an adult with an IQ of 70, what's the benefit of diagnoses if they're seemingly functional? It would be over 70. And I know 70 is what our funders use for the PDDH funding. My response to that is, how are they functioning? What supports are they receiving to date? I think the snapshot of that particular adult, they've probably not been able to keep employment. They've probably not been really good at managing their money. They've probably had troubles accessing appropriate housing without supports. So I think the advantage would be to have the evidence of brain damage. The IQ does not define this disability. In fact, we often look at the IQ level is kind of our threshold, and all the other testing that we do is down below. So that's where IQ does not define this disability. And if that individual is functioning, I bet they've got their peeps. I bet they've got their support team surrounding them. And I actually, I've met some one person I'm thinking of is a fabulous artist. He presented at the BC FASD conference. His wife is fantastic. She manages money, booking his art shows, making sure all the other things. And he's creative and he's functional and successful. His wife is the external brain.
Thank you very much, Gail. As Shelley Burchard here again, I realized that I didn't introduce myself. I was so focused on introducing Gail here today. As the co-chair of the FASD CMC, I'm taking away two important messages from this presentation as opportunities to focus our work for the FASD 10-year strategic plan: prevention of future affected births and long-term lifespan supports to individuals identified with FASD. There's much information that was shared today, much valuable information. I'm extremely pleased at the number of registrants that we had, and I'm hopeful that you all found this webinar informative, and that each of you may take the step to sharing this information with your families, friends, colleagues. This video and PowerPoint will be available on FASD website at www.fasd-cmc.ab.ca. For people that have asked for resource references, these can be found in the handout and downloaded from the PowerPoint website. Thank you to every one of you who shared their questions. For those, much like Gail just mentioned, that for the inquiries that you still have that were left unanswered, by all means, please contact her, which I believe is the information on the last page of the PowerPoint. And also, thank you for all the positive comments that the webinar seemed to fulfill needs out there, which is great. So again, thank you for your time, for tuning in. I welcome you guys to share this information, have other people download the webinar at different times in the future. And again, thank you, Gail, for your time and energy today to share the valuable information.